Today in this lecture, we will talk about the antidiuretic hormone synthesis and its release and its connection with different areas of the brain in maintaining concentration of sodium in plasma. This lecture is in continuation with or the last lecture, the osmoreceptor antidiuretic hormone pathway or mechanism. Now we know that to make concentrated or dilute urine, the kidneys are very much dependent upon the concentration of plasma, and plasma concentration is very much dependent upon the concentration of sodium, and sodium concentration is maintained to a very big extent by the antidiuretic hormone, and the release of antidiuretic hormone is again dependent upon the plasma osmolarity. So how this cycle works? Now we have discussed previously that in the hypothalamus there are areas known as supraoptic and paraventricular areas, and these supraoptic and paraventricular areas, they are making antidiuretic hormone. Around 1/6 of the antidiuretic hormone is made in the periventricular area, and around 5/6 is made in the supraoptic nuclei. And this antidiuretic hormone, once synthesized in these areas, it is transported through the neurons into the posterior pituitary gland. This is the posterior pituitary. This is the anterior pituitary. Now, the supraoptic and paraventricular areas, they are connected with the osmoreceptors. Osmoreceptors, they detect the concentration of sodium in plasma, or they detect the plasma osmolarity. And if there is a high increase in plasma osmolarity, if there is increase in plasma osmolarity due to increase in sodium concentration, it will get activated and it will lead to release of antidiuretic hormone from posterior pituitary it will lead to release of ADH into blood, and this ADH will then act on the kidneys and lead to reabsorption of water and correction of plasma osmolarity. Now, not only is the release of this ADH from posterior pituitary dependent upon the signals coming from the osmoreceptors in the hypothalamus, but it is also dependent on another stimulation of another area that is known as AV3V region. Now, this AV3V region is present in the anterior ventricular region of the third ventricle. So that's why it is known as AV3V region, present in the anterior ventricular area of the third ventricular. Now, we will not go into the detail of neuroanatomy, but just remember that the name of this region is AV3V area because it is located in specific part of the third ventricle. Now, in this AV3V region, in the upper part we have the subfronical organ. This is the subfronical organ. In the lower area we have the organum vasculosum, which is part of the lamina terminalis, and in the middle of the AV3V region we have the median preoptic nucleus. Signals from the subfronical organ and organum vasculosum are going to the median preoptic nucleus, and this median preoptic nucleus is also receiving signals from the centers of brain which are controlling the blood pressure. Now, this median preoptic nucleus is receiving signals from here, there, and from blood pressure center, and now it is also connected with the supraoptic nuclei. So the release of antidiuretic hormone is not only dependent upon stimulation of supraoptic and paraventricular nuclei with the help of osmoreceptors present in the hypothalamic region, but it is also dependent upon the activation of this AV3V region, which is further connected with different areas. So not only the changes in the osmolarity, not only the chemical changes, but also changes in the blood pressure, the arterial pressure, will lead to release of antidiuretic hormone. Now, if this area is disturbed, if there is a lesion in this AV3V region, there will be abnormalities of antidiuretic hormone release, there will be abnormalities of thirst, there will be abnormality of sodium appetite, and there will be abnormalities of blood pressure control, because this area is very much important in the release of antidiuretic hormone, which again is very much important for the reabsorption of water, and reabsorption of water is very much important for the control of arterial pressure or blood pressure. Now, why would any lesion of this area lead to these abnormalities? Because normally the AV3V region is stimulated. It is stimulated by changes in the thirst, sorry, this area normally stimulate the thirst. This region stimulates sodium appetite, and this area stimulates control of blood pressure through release of antidiuretic hormone. So any change in the blood pressure, any change in the arterial pressure, or any change in the plasma osmolarity will be detected through these areas, and it will be communicated to the hypothalamus, and then hypothalamus will lead to release of antidiuretic hormone. And once the antidiuretic hormone is released into the blood, it will go into the kidneys, and it will lead to reabsorption of water and correction of the abnormality. Now, it is important to note that some osmoreceptors are present in this area as well, around the subfronical organ and the organum vasculosum. And this subfronical organ and organum vasculosum, these areas have no special blood-brain barrier, no special blood-brain barrier. So the changes, the chemical changes, are detected very easily by the subfronical organ and the organum vasculosum. Normally, when there is a blood-brain barrier, suppose for example this is a blood vessel and this is the blood-brain barrier, the chemical changes cannot directly come into contact with the brain area because of the blood-brain barrier. But in these areas, the blood-brain barrier is interrupted, or there is no specific blood-brain barrier, which lead to easy communication of changes in the osmolarity, or easy communication of the chemical changes with the organum vasculosum subfronical area, and ultimately the medial preoptic nuclei. So that's all about the ADH synthesis and its release and its connection with different areas of the brain, and its relation with the ADH osmoreceptor mechanism. In the next lecture, we will discuss that how the ADH is affected by the changes in the chemical changes and the physical changes in the blood pressure. So that's all about the ADH synthesis and its release through different pathways. Thanks a lot for watching the video.